Hello everyone, I'm Teacher Vien and welcome to Biology class. Our lesson for today is about cellular organelles. But before we get started, let me introduce to you our learning competency. At the end of this video lesson, you shall be able to describe the structure and function of major and subcellular organelles. Now let's begin. Let's talk about cellular organelles, structure, and function. A cell is the smallest unit of a living thing. A living thing which can be made up of one cell, just like the bacteria, or many cells, just like a human, is called an organism. Thus, cells are the basic building blocks of all living organisms. Most cells are so tiny that we cannot see them using our naked eye. Therefore, scientists use microscopes to study cells. Have you ever heard this phrase? Form follows function. It's a philosophy practiced in many industries. In architecture, this means that buildings should be constructed to support the activities that will be carried out inside them. For example, a hospital should be built so that its emergency room is easily accessible. Our natural world also utilizes the principle of form following function, especially in the study of cells. And this will become clear as we explore eukaryotic cells, because a eukaryotic cell's nucleus is enclosed by a membrane. It is often said to have a true nucleus. The word organelle means little organ, and organelles have specialized cellular functions, just as the organs of your body that have specialized functions. In this lesson, you will learn a variety of structures and functions of major and subcellular organelles. Let's take a look at this sample cell. It is a eukaryotic cell, particularly an animal cell. It has true nucleus and membrane-bound organelles. Another eukaryotic cell is the plant cell. We're going to compare plant cells and animal cells in our next video lesson. But for now, let's discover structures and functions of a cell. Let's discuss the nucleus and the ribosomes. The nucleus contains most of the cell's DNA, its master plans, and controls the cell's activities by directing protein synthesis. The DNA is associated with many proteins in the structures called chromosomes. The proteins help organize and coil the long DNA molecule. When a cell is not dividing, this complex of proteins and DNA, called chromatin, appears as a diffuse mass. Enclosing the nucleus is a double membrane called the nuclear envelope. The nucleolus, a prominent structure inside the nucleus, is the site where a special type of RNA called ribosomal RNA is synthesized according to the instructions in the DNA. Let's answer this question. What are the main functions of the nucleus? You're correct! To house and copy DNA and pass it on to daughter cells in cell division, to build ribosomal subunits, and to transcribe DNA instructions into RNA and thereby control the cell's functions. Let's move on to the ribosomes. If the nucleus is the command center, then ribosomes are the machines on which those commands are carried out. ribosomes synthesize proteins. The nucleus directs protein synthesis by making another type of RNA, which is the messenger RNA. Essentially, mRNA is transcription of protein synthesizing instructions written in a gene's DNA. The mRNA moves through the pores in the nuclear envelope to the cytoplasm. There, it is translated by ribosomes into the amino acid sequence of proteins. Let's answer this question. What role do ribosomes play in carrying out the genetic instructions of a cell? Exactly! Ribosomes synthesize proteins according to the instructions carried by mRNA from the DNA in the nucleus. Many of the membranes of the eukaryotic cell are part of an endomembrane system. The endomembrane system includes the nuclear envelope, endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, and the plasma membrane. Now let's study the endoplasmic reticulum. One of the major manufacturing sites in a cell is the endoplasmic reticulum. It is a biosynthetic factory. There are two types of ER, the smooth ER and the rough ER. Smooth endoplasmic reticulum is called smooth because it lacks attached ribosomes, while rough ER has ribosomes that stud the outer surface, thus it appears rough in the electron micrograph. Enzymes of the smooth ER are important in the synthesis of lipids, including phospholipids, oils, and steroids. In vertebrates, for example, cells of the ovaries and testes synthesize the steroid sex hormones. These cells are rich in the smooth ER, a structural feature that fits their function by providing ample machinery for steroid synthesis. One of the functions of rough ER is to make more membrane. Phospholipids made by enzymes of the rough ER are inserted into the ER membrane. Thus, the ER membrane grows, and portions of it are transferred to other components of the endomembrane system as vesicles. The bound ribosomes attached to rough ER produce proteins that will be inserted into the growing ER membrane, transported to other organelles, or secreted by the cell. An example of a secretory protein is an insulin, a hormone secreted by specialized cells in the pancreas. Type 1 diabetes results when these cells are destroyed and a lack of insulin disrupts glucose metabolism in the body. The diagram on the right side follows the synthesis, modification, and packaging of a secretory protein. Now let's answer this question. Explain why we say that the endoplasmic reticulum is a biosynthetic factory. You're correct! The ER produces a huge variety of molecules, including phospholipids for cell membranes, steroid hormones, and proteins for membranes, other organelles, and secretion by the cell. Now let's explore the Golgi apparatus. After leaving the ER, many transport vesicles travel to the Golgi apparatus. The Golgi apparatus serves as a molecular warehouse and finishing factory for products manufactured by the ER. You can follow this process in the figure. Note that the flattened Golgi sacs are not connected, as are ER sacs. One side of a Golgi sac serves as a receiving duct for transport vesicles produced by the ER. A vesicle fuses with a Golgi sac, adding its membrane and contents to the receiving side. Products of the ER are modified during their transit through the Golgi. The other side of the Golgi, the shipping side, gives rise to vesicles which bud off and travel to other sites. Now let's answer this question. What is the relationship of the Golgi apparatus to the ER in a protein-secreting cell? Excellent! The Golgi receives transport vesicles that bud from the ER and that contains protein synthesized by ribosomes attached to it. The Golgi finishes processing that proteins and then dispatches transport vesicles that secrete the proteins to the outside of the cell. Next structures are the lysosomes. Lysosomes are digestive compartments within a cell. The name lysosome is derived from two Greek words meaning breakdown body. Lysosomes have several types of digestive functions. Many protists engulf food particles into membranous sacs called food vacuoles. As the figure shows, lysosomes fuse with food vacuoles and digest the food. The nutrients are then released into the cell fluid. Our white blood cells engulf and destroy bacteria using lysosomal enzymes. Lysosomes also serve as a recycling center for animal cells. Damaged organelles or small amounts of cell fluid become surrounded by a membrane. While another lysosome fuses with such a vesicle in this figure and dismantles its contents, making organic molecules available for reuse. With the help of lysosomes, a cell continually renews itself. Please take note that we also have what we call as peroxisomes, which are metabolic compartments that do not originate from the endomembrane system. In fact, how they are related to other organelles is still unknown. Some peroxisomes break down fatty acids to be used as cellular fuel. In your liver, peroxisomes detoxify harmful compounds, including alcohol. In these processes, enzymes transfer hydrogen from various compounds to oxygen, producing hydrogen peroxide. Now let's answer this question. How is a lysosome like a recycling center? Bravo! It breaks down damaged organelles and recycles their molecules. We also have structures called vacuoles. Vacuoles are large vesicles that have a variety of functions. In the left image, you see how a food vacuole forms as a cell ingests food. The image shows two contractile vacuoles in the protist paramecium, looking somewhat like wheel hubs with radiating spokes. The spokes collect water from the cell, and the hub expels it to the outside. Freshwater protists constantly take in water from their environment. Without a way to get rid of the excess water, the cell would swell and burst. See the right image. It shows a plant cell's large central vacuole, which helps the cell grow in size by absorbing water and enlarging. It also stockpiles vital chemicals and acts as a trash can, safely storing toxic waste products. Let's answer this question. Is a food vacuole part of the endomembrane system? It's a yes! It forms by pinching in from the plasma membrane, which is part of the endomembrane system. Now let's talk about these energy-giving organelles, which are the mitochondria. Mitochondria, or mitochondrion in singular form, are organelles that carry out cellular respiration in nearly all eukaryotic cells, converting the chemical energy of foods, such as sugars, to the chemical energy of the molecule called ATP, or adenosine triphosphate. ATP is the main energy source for cellular work. We will discuss the role of mitochondria in more detail as we discuss the cellular respiration on the second quarter of General Biology 1. Now let's answer this question. What is cellular respiration? You're correct! Good job! Cellular respiration is a process that converts the chemical energy of sugars and other food molecules to the chemical energy of ATP. Now let's talk about chloroplast. Chloroplasts convert solar energy to chemical energy. Most of the living world runs on the energy provided by photosynthesis, the conversion of light energy from the sun to the chemical energy of sugar molecules. Chloroplasts are the photosynthesizing organelles of all photosynthetic eukaryotes. In photosynthesis discussion, we're going to discuss this in detail. But for now, let's answer this question. Are chloroplasts present in animal cells? You're correct! It's a no! Chloroplasts are present in the cells of all green tissues of plants and algae. Other cell structures include the cytoskeleton and cell surfaces. Biologists once thought that organelles floated freely in the cell, but improvements in both light microscopy and electron microscopy have allowed scientists to uncover a network of protein fibers, collectively called the cytoskeleton, extending throughout the cytoplasm of a cell. These fibers function like a skeleton in providing for both structural support and cell motility. There are three main kinds of fibers that make up the cytoskeleton. One is the microfilaments, the thinnest fiber. Next, we have microtubules, the thickest. And third, we have intermediate filaments in between the thickness. Microfilaments, also called as actin filaments. They are solid rods composed mainly of globular proteins called actin, arranged in a twisted double chain. Microfilaments form a 3D network just inside the plasma membrane that helps support the cell's shape. This is especially important for animal cells, which lack cell walls. Intermediate filaments may be made of various fibrous proteins that supercoil into thicker cables. They serve mainly to reinforce cell shape and to anchor certain organelles. The outer layer of your skin consists of dead skin cells full of intermediate filaments made of keratin proteins. Microtubules are straight, hollow tubes composed of globular proteins called tubulins. As indicated in the bottom right of the figure, microtubules elongate by the addition of tubulin proteins, which consist of two subunits. Microtubules shape and support the cell, and also act as tracks along which organelles equipped with motor proteins move. For example, a lysosome might walk along a microtubule to reach a food vacuole. Microtubules also guide the movement of chromosomes when cells divide. And as we see next, they are the main components of cilia and flagella, the locomotive appendages of cells. Now let's answer this question. Which component of the cytoskeleton is most important in A. Holding the nucleus in place within the cell, B. Guiding transport vesicles from the Golgi to the plasma membrane, and C. Contracting muscle cells? Great! The answer for letter A is intermediate filaments. For letter B, we have microtubules, and for letter C, we have microfilaments. Let's move down to the last two structures, which are the cilia and flagella. The role of the cytoskeleton in movement is clearly seen in the motile appendages that protrude from certain cells. Some cells of multicellular organisms also have cilia or flagella. For example, the left image shows cilia on cells lining the human windpipe or trachea. In this case, the cilia sweep mucus-containing trapped debris out of your lungs. Most animals and some plants have flagellated sperm. A flagellum, shown in the right figure, propels the cell by an undulating whip-like motion. In contrast, cilia work more like the coordinated oars of a rowing team. Now let's answer this question. Compare and contrast cilia and flagella. You got it! Cilia are shorter, are more numerous, and beat in a coordinated oar-like pattern, while flagella is longer, which are limited to one or a few per cell, undulate like a whip. Now, let's summarize what we have discussed about cellular organelles. Eukaryotic cells have different structures that help them to function. Some of those structures are nucleus, peroxisome, endoplasmic reticulum, cytoskeleton, plasma membrane, lysosome, Golgi apparatus, cytoplasm, mitochondria, vacuole. There are also other parts that can be seen among plant cells, which are plasmodesmata, cell wall, central vacuole, and plastids. Thank you for watching this visual lesson, and I hope to see you on our next visual lesson. Bye!